Now we'll start with the chapter 6. Chapter 6 is about the gastrulation and the genes that will control the formation of axis in vertebrates. So this chapter is divided into two parts. The first part that I'm going to talk about today, it's a kind of introduction. I will remind you about the different stages of development that happens usually in human. And in the second part, we'll talk about gastrulation and how genes can control the formation of the different body axes. So you know that usually the embryo, when it's implanted at the level of the endometrium of the uterus, it's called blastocyst. Okay, so the blastocyst will be implanted, and after the implantation of this blastocyst, okay, which is the equivalent of the blastula in sea urchin or in frogs, for example, the blastocyst, it's called blastocyst in mammals or in human. Okay, so we'll talk about the first week, what happens after the first week of the blastocyst implantation, and the second week. In the second part of this chapter, we'll start with the third week of development, which is the week of gastrulation. So gastrulation starts by the third week of development. But before the gastrulation, we'll talk about the main events happening during the first and the second week of development. Okay, so by gastrulation, or by the beginning of the third week, we'll have migration of the cells, okay, or ingression of the cells, that I will talk about it in detail in the second part of this chapter, by the process of the gastrulation in order to form a trilaminar germ disc. It means this embryo will look like a disc formed by 3 embryonic layers. That's why it's called trilaminar germ disc. By the second week of development, we talk about a bilaminar germ disc, okay, that I will show you this embryo, how it looks like. So we have only 2 embryonic layers, so we have only ectoderm and endoderm. Here, by the third week, by gastrulation, we have a trilaminar germ disc, so we have the mesoderm that will be formed between ectoderm and endoderm. So here you have a link, so you can open it through YouTube and you can watch this different events of the first week of development, and you already know them from the animal reproduction course in the first year. So you know that after ovulation you have the fertilization by the sperm. Okay, if it's the case, we'll have a zygote, a diploid cell that will divide into 2 blastomeres or 2 cells just 30 minutes after fertilization. Okay, and just after we'll have 4 cells, 8 cells, then the morula stage, and after the blastocyst. Okay, the blastocyst is the stage or the embryonic stage that will be implanted at the level of the endometrium. Okay, and this takes place usually by the day 6 after fertilization, or even by the end of the first week of embryonic development. So these events usually happen in the first week of development. By the second week of development, as I mentioned before, we'll have a bilaminar germ disc. It means the embryo will be formed by only 2 layers. That's why it's called bilaminar, so ectoderm and endoderm. So here in the second week, we'll take the critical days of development. Through these days, we'll have main events of embryonic development. So we'll talk about the day 8, it means by the beginning of the second week, the day 9, what are the events happening by the day 9, the days 11 and 12, since we have the same things happening during these days, and finally the day 13 that describes the end of the second week of development. You also have a link that you can open it, and if you watch this video, you will see the main events happening during these 4 days. Okay, so it's a very nice video, you can watch it as well. But I will explain the main things described in this video in the next slides. So this is the blastocyst. The blastocyst is formed by an inner cell mass called embryoblast. It's the part of the embryo that will give the different embryonic layers: ectoderm, mesoderm, endoderm. And we have this outer layer, outer cell mass. It's called trophoblast, that will give the placenta. Okay, this is the embryonic pole where the embryoblast is present, and this is the opposite pole called abembryonic pole. So here in this figure, you see that it's the beginning of the day 8. Okay, you see this is the human blastocyst, okay, which is partially embedded in the endometrium. This is the surface of the endometrium, and usually the endometrium is a line or covered or protected by one columnar layer of epithelial cells. We have the surface epithelium. It's a protective layer surrounding the endometrial stroma. So in order to penetrate, the blastocyst should be penetrating between these epithelial cells. Okay, so in order to be embedded inside at the level of the endometrium. So by the day 8, the blastocyst, you see in this figure, it's still partially embedded. Okay, it's partially embedded in the endometrium. And you see, this is the embryoblast that I talked about before. It has been differentiated by the beginning of the second week, so by the day 8, into 2 layers. We have the amnioblast. This layer is formed by columnar cells. Okay, I'm talking about the layer in blue. It's formed by columnar cells adjacent to a small cleft, okay, representing the amniotic cavity, which is still very, very small or narrow at this stage of development. The second layer is just below the epiblast. Epi, it means above. That's why this layer is called epiblast. It will give in the future the ectoderm. And below we have the hypoblast. Hypo means below, and it will give the endoderm. This is the embryoblast, so it has been differentiated into epiblast and hypoblast. So over the area of the embryoblast, the trophoblast has been also differentiated into 2 layers: the cytotrophoblast, this one, the cytotrophoblast, the inner layer, and the syncytiotrophoblast, the outer layer. The cytotrophoblast is a mononucleated layer, while the syncytiotrophoblast, it's a zone, it's a multinucleated mass, since the nuclei has been fused together to form this kind of layer called syncytiotrophoblast. If you remember, the syncytium in Drosophila was formed by one cell with many nuclei. Here it's the same. That's why it's called, this layer is called syncytiotrophoblast. So since the amniotic cavity is present right here, the epiblastic cells just adjacent to the cytotrophoblast, okay, in front of this amniotic cavity, are called amnioblasts. Just these ones, okay, these are called these are epiblastic cells, but in detail we call them amnioblasts. So by this stage we can see that we have the uterine glands, okay, that can secrete the mucus. Okay, they can also secrete abundant amounts of glycogen, and also we have the blood vessels, or we can see that the endometrial stroma adjacent to the site of implantation of the blastocyst is highly vascular.